All right, today we're going to be talking about how eukaryotic cells form via endosymbiosis. And I'm going to go pretty quick, but you can always come back and watch this video and pause to give yourselves more time to write or to double check some of these facts. Here's your learning targets. And let's start off with just by talking about when first cells evolved. So we should know that protocells were around about 3.8 billion years ago. We know they were really simple cells. We know there were prokaryotic cells. And for a long time, about 2 billion years after that, nothing happened. So they might have gotten a little more complex. They might have been able to have a little more complex function, slightly more complex structure. But essentially, we had prokaryotes on our planet for a really long time. And it wasn't about till 1.8 billion years ago that eukaryotic cells formed. And that's what we're going to talk about today, is how that happened. The two processes that are thought to have led to the origin of eukaryotic cells are called infolding and endosymbiosis. So we're going to discuss which organelles or which structures were made via infolding and which ones were made via endosymbiosis. So let's start with infolding. Infolding is pretty straightforward, and you can see this little GIF that will just keep repeating in the upper right-hand corner of the screen. But essentially, we have organelles like the endoplasmic reticulum, so both of them, smooth and rough. The Golgi apparatus and the nuclear envelope are thought to have evolved from this process called infolding, which is essentially when the plasma membrane of our really ancestral basic protocells or prokaryotes started to fold in on itself, just like you can see starting to happen in this kind of second picture. So we have our ancestral prokaryote, and then the cell membrane starts to fold in on itself. And you might ask yourself, like, why would it do that? Well, we think that the increase in surface area was the evolutionary benefit to potentially why that was a beneficial trait. So if you guys think about this, if I add more folds, what I'm actually adding is more surface area. And we've briefly talked about how a lot of reactions actually happen in the membrane itself. So if I'm trying to have, or if I'm an organism and efficiency is essential to my survival, it would make sense that the more surface area I have, the more efficient I can be. So that's kind of why we think infolding was a successful adaptation. And from that, we think those infolds basically broke off and maintained inside the cell, and over time developed different structures and functions of their own. So like, for example, in this little GIF, we can see that the infolding of the cell membrane actually then breaks off and joins together to form the nuclear membrane and starts to form those compartments that we have talked about. So that's the gist of infolding. And we think these internal membranes basically would allow the cell to carry out more and more complex reactions in these separate compartments via compartmentalization, which is one of the things we've talked about. So once again, things like the endoplasmic reticulum, which is basically just folded cell membrane, right? Same thing with the Golgi apparatus, folded cell membrane. And so this process of infolding hopefully makes sense. Okay, endosymbiosis is another step in the evolution of eukaryotic cells. And what we think happened is the two organelles that we think for sure evolved via endosymbiosis are chloroplasts and mitochondria. And what we think is they actually used to be smaller prokaryotic cells that evolved to live within their host cells, and then they formed a symbiotic relationship. So before we go any farther, we should define this word symbiosis. Technically, symbiosis is just an interaction between two different organisms, like two different species that live in close physical association. There's different types of symbiosis, and the type that we're talking about today is actually called mutualism. And mutualism is when both organisms benefit, as opposed to something like parasitism, which you guys have probably heard of before, is actually another type of symbiosis. But if you know what a parasite is, parasites are usually benefiting while the host is not benefiting. So that would be an example where one organism benefits, but the other one is actually being harmed. So any type of relationship where the organisms live closely together is called symbiosis, and mutualistic symbiosis is when both organisms mutually benefit. So in endosymbiosis, that's when one cell lived within the other and became increasingly dependent on each other, actually interdependent on each other, until they could only exist as a whole unit. So let's talk a little bit more about this process. Starting with mitochondria. So we think that the first symbiotic ancestors of mitochondria were actually aerobic bacteria that did cellular respiration, right, using oxygen. And at the time, they lived by themselves. They were simple cells that could turn food particles into energy. At some point in time, what we think happened is they got ingested by a larger host cell. And this host cell might have been slightly more advanced. Maybe it looks like, based on our picture, right, that the infolding process had already happened and begun. So these were larger cells. They were slightly more complex. They were already starting to compartmentalize. And at that point, for some reason, this is the part we're not entirely sure why, the mitochondria didn't get, well, the early mitochondria, this prokaryote, didn't get digested, because normally we've learned via like phagocytic cells that if a cell ingests another cell, like a bacterium or a pathogen, usually those digestive enzymes break it down. But for some reason, this cell didn't digest it, and it actually remained alive inside the host cell and continued to do its job, which is cellular respiration. So from that, both cells actually ended up benefiting. So I'm hoping you're thinking to yourself, how did the host cell benefit? And how did this like pre-mitochondria benefit? Well, if you think about it for a second, I would hope you would think that if the mitochondria is making energy, that cell could harness that energy. And then as the host cell, I would think, maybe structure or support or protection or food source. So those are kind of the reasons that we think the host cell would actually benefit and not potentially break down this little aerobic bacterium. So that's kind of the thought on the mitochondria evolution. We think something really similar happened with chloroplasts. So chloroplasts were photosynthetic bacteria that, once again, before all this happened, lived outside of a larger host cell. But then for some reason, once again, was brought inside of a host cell. The host cell was going to digest it. And for some reason, it didn't. So these photosynthetic bacteria actually remain alive, start to form a symbiotic relationship with a host cell, and ended up living inside the host and thriving. So if you're thinking to yourself, hopefully, what kind of cell would have a symbiotic relationship with a chloroplast? You're right. Plant cells, right? So this is essentially how plant and animal cells started to diverge. And none of this would be possible without the structure of the cell membrane. So we should remember that the fluidity of the cell membrane is what allows so much of this to happen, right? It's essential to our endomembrane system. It's essential to the endosymbiosis theory. It's essential to infolding. So we really truly think this is how a lot of these things evolved. And because all cells, all eukaryotic cells have mitochondria, we think mitochondria endosymbiosis happened first. And then because only some cells have chloroplasts, we think the evolution of chloroplasts happened second. But the process is very similar, and this is one of the big diverging points between plant cells and animal cells. So when I say endosymbiosis theory, I just want to emphasize one more time, we're not using the word theory like we would in general conversation. Like, hey, I have a theory why Ben and Susie broke up, right? That's a very different conversation. So if I say endosymbiosis theory in science, I mean this is a well-substantiated, AKA lots of evidence, explanation of the natural world based on a body of facts, not opinion-based, repeatedly confirmed, and then we can see it or prove it through observation, modeling, experimentation, etc. In fact, a pretty famous woman in cell world came up with the endosymbiotic theory, or was a big part of it, Lynn Margulis, and she died in 2011. But yay for women in science. We love it. So hopefully at this point, you're asking to yourself, why should I believe this? Like, what's the evidence behind endosymbiosis? So let's go through and talk about 3 different kinds of endosymbiosis, or 3 different pieces of evidence. And I think you'll start to kind of see that the body of evidence together makes this a very strong theory. So let's start off with structural evidence. First off, mitochondria and chloroplasts are just similar in size and shape to many prokaryotes. So that's a pretty simple one. And by itself, size and shape is not enough to convince me, right? Like, so if that's all the evidence I had for endosymbiosis theory, I would dismiss it. So let's keep going. On top of that, we know that mitochondria and chloroplasts are 2 of our organelles that have a double membrane. I don't know if you ever stopped to think, how would a double membrane form? Or why would a double membrane form in some organelles and not in others? And the theory of endosymbiosis actually supports this. So if you watch the GIF on this screen, you'll see that as like this early mitochondria is ingested by the cell, right? In the picture, they're labeling it as a bacterium, but we know this is like a pre-mitochondria. Essentially, a little vesicle forms around it. And as the vesicle forms around it, we know that vesicle is made out of, you're saying, phospholipids. So this is actually how the second membrane, the outer membrane, forms. This is the theory, at least, is that the engulfed membrane, or the engulfed bacteria, now has a double membrane around it, because as it was getting engulfed, it got put in a vesicle. So those are good 2 structural bits of evidence. Let's do one more. Another one is the ribosomes. You may or may not know it, but mitochondria and chloroplasts both have ribosomes. And not only do they have ribosomes that are similar to prokaryotes. So we should remember that eukaryotes have 80 S ribosomes. And so structurally, they're a little different in size. But the fact that chloroplasts and mitochondria, or at least the early stage ones, right, and actually current ones too, have ribosomes, tell me that these potentially at one point in time were fully functioning organisms by themselves, right? Because remember, we said all cells, all living things have ribosomes because ribosomes make proteins and we cannot survive without proteins. So the fact that they have ribosomes and they're similar to prokaryotic ribosomes is just another piece of structural evidence. Okay, genetic evidence. So this one is also pretty compelling. Both mitochondria and chloroplasts have circular DNA. Circular naked DNA. And that's a phrase you guys should know at this point. And that is exactly what prokaryotes have. So remember, eukaryotes have linear DNA. They have DNA that's associated with histone proteins. So the fact that this DNA is structurally very similar to prokaryotes, kind of another piece of evidence to add to our growing list. And on top of that, we know that in this class, we've learned a very good phrase, technology begets discovery. We know that with modern day science, we've been able to do some DNA sequencing of chloroplast DNA, mitochondrial DNA, and we can actually see that there are some common genes or common DNA sequences between those organelles and prokaryotes. So it's not just structural, it's also at like the core of what these molecules are. So that's pretty cool. And then our last piece of evidence is what we're titling functional evidence. So functional evidence, both mitochondria and chloroplasts can move independently within the cell, right? And we know that prokaryotes can move within the cell or move by themselves, often with the help of one of my favorite proteins, which you guys should know by now. And then another example is they can reproduce independently, meaning that they don't need the cell to be going through cell division. They actually can reproduce on their own. So let's say a cell is very energy dependent, like your heart muscle cells. They probably have a lot of mitochondria. So they actually might create more mitochondria as your heart develops and needs more energy. So it's pretty cool that these guys can reproduce in a process that's really similar to how prokaryotes reproduce, which is what we call binary fission. The last one, and I think this one is the coolest, is the fact that we know, hopefully you guys know what antibiotics are. We've talked a little bit about these. What's weird and interesting is chloroplasts and mitochondria can actually be inhibited or stopped. Their functions can be shut down with the use of antibiotics. And if you know what an antibiotic does, we know antibiotics are designed to shut down or inhibit bacteria. So the fact that they would respond in the same way as a bacterium, one more piece of evidence that tells me I am looking at something that is probably a prokaryotic organism billions of years ago, which has now merged with our modern day eukaryotic cells through the process of endosymbiosis. And there's one more example, hang tight. The front of your textbook actually has a photosynthetic sea slug. So in a nutshell, I'm not going to read this to you word for word, but you can read this little synopsis on the back. In a nutshell, this photosynthetic sea slug has ingested algal cells, and that has allowed them to become photosynthetic themselves. So these sea slugs can actually not eat for like 2 to 3 months at a time and get energy from their photosynthetic symbiotic counterparts, which is super, super cool. So this isn't just something that happened like billions of years ago. At the end of this Amoeba Sisters video, which we're going to watch in a second, they'll also give you a few more examples of how endosymbiosis has happened. So that's about all. I'm going to pause the video here for copyright. It won't let me record Amoeba Sisters. So you guys are going to watch Amoeba Sisters separately, but hopefully this was helpful and good luck.